Good morning.
Good morning.
Good morning. Okay, so now we will start with biotechnology, with vectors, with cloning, and with PCR. So, vectors, cloning, and PCR are important techniques in molecular biology and biotechnology. These techniques are essential for gene manipulation, which is a core aspect of genetic engineering, gene therapy, and various research applications. Let's start with the vectors. Vectors as a definition, they are DNA molecules that are used to carry foreign genetic material into another cell where it can be replicated or expressed. The vector itself must have the ability to replicate within a host cell, ensuring that the inserted genetic material is also replicated. So the vector itself, it must need, like any virus, like the virus, it must need a host cell to be able to replicate. It needs a space, it needs a place to start replicating. However, the vector is different from the virus, that when this vector comes and takes a side to start replicating, DNA, the free DNA inside this vector will also replicate, will become 2Q. Okay? DNA material. What are the vectors? Vectors are DNA molecules that are used to carry foreign genetic material into another cell. Okay? So, so what are the types of vectors? Types of vectors. We have the plasmids. We have the bacteriophages. The cosmids. The BACs. The YACs. And the viral vectors. Okay? So there are 6 types of vectors. What are the plasmid vectors? Plasmid vectors, or the plasmids, what are they? They are circular DNA molecules found in bacteria. They are the most commonly used vectors. Plasmids often contain an allergen of replication, a selectable marker. What is the selectable marker? Selectable marker is mainly antibiotic resistance genes. So selectable marker gene. So what are plasmids? Plasmids are the most known type of vectors. They look like this. They are circular DNA molecules found in bacteria. They are most commonly used vectors, and they contain an origin of replication, a selectable marker, and multiple cloning sites, which is called MCF, multiple cloning sites, where the DNA of interest can be inserted. So the plasmids are found in bacteria. They are circular DNA. And as we said, we spoke about it before when we were speaking about the bacteria, the prokaryotes, or we spoke about the E. coli bacteria, which looks like this. We said it has circular DNA since it's a prokaryote. So this circular DNA itself, around it, since it does not have a nucleus, it's just inside a region which is called the nucleoid. This means that DNA can be going out of this DNA freely because there's nothing or there's no layer which is surrounding this region. So here DNA, with the movement of these E. coli, with the replication of this E. coli bacteria, this DNA is also moving inside in this cytoplasm. And this DNA will start to go out as vesicles and circular vesicles, which are called the plasmids. What are the plasmids? Are free circular DNA around the DNA or around the circular DNA inside the bacteria. Another type of plasmids are the vectors are the bacteriophages. Viruses that infect bacteria. So the bacteriophages are mainly the viruses that infect bacteria. Okay. The bacteriophages come from a word which is called the phage vectors. Like the lambda phage, we have the phage vectors. As an example, the lambda phage. What does this phage vectors do? They can carry larger DNA fragments than plasmids. So they can carry larger DNA fragments than plasmids. So the only difference between bacteriophages and normal plasmids is that they can carry larger DNA fragments than them. As an example of the bacteriophages and of the phage vectors, we have the lambda phage. Another kind of vectors are the cosmids. What are the cosmids? Are hybrid vectors that combine features of plasmids and bacteriophages. So it's a kind of combination between the plasmids and the bacteriophages. Okay? Allowing for the cloning of larger DNA fragments. So it's a kind of hybrid vector. Hybrid means more than one. Combination between plasmids and bacteriophages. Okay. What does it help? It helps in the cloning of large DNA fragments around or up to 45 kb. What are the BACs? The BACs are also called the bacterial artificial chromosomes. Yeah. These bacterial artificial chromosomes are large vectors based on a functional fertility plasmid, which is called the F plasmid. So they base on this functional fertility plasmid, which is called the F plasmid. Okay. They used to clone DNA fragments between 100 and 300 kb, means very huge molecule of DNA fragment. We must use the BAC. It's artificial because we won't have in nature this kind of plasmids that's able to clone this big DNA fragments. However, the YACs are the yeast artificial chromosomes. So that one is the bacteria. Here we have the yeast artificial chromosomes. Okay. These are vectors used to clone very large DNA fragments up to 1 MB in yeast cells. So this is specific for yeast. The last kind of vectors that we must know are the viral vectors. The viral vectors are used in gene therapy to deliver genetic material into human cells. What are examples of viral vectors? So let's write first the definition. They are used in gene therapy to deliver genetic material into human cells. Example, adenoviral vectors, lentiviral vectors, and retroviral vectors. So these are used in gene therapy to deliver this DNA material inside the human. Okay. Now, let's see what are the vector components. So, the vector components are the ORI, are the marker genes, MCS, and the promoter. These are the main components of the vector. The ORI is the origin of replication. What is the ORI? It's the site where DNA replication begins, allowing the vector to replicate within the host cell and to replicate its DNA material. Selective marker genes. These genes allow the identification of cells that have taken up the vector. What are the kinds of markers? What are the marker genes? Marker genes are like, as we said, the antibiotic resistance genes. As an example, we have the ampicillin, licannamycin, etc. So, origin of replication is the main site where DNA replication begins. Selectable markable genes are the antibiotic resistance genes, the MCS, which are called the multiple cloning sites, It's a region containing several restriction enzymes. What are the restriction enzymes? Are the enzymes that try to cut the DNA, try to cut the chromosome. Of course, it does try to cut the chromosome, but of course not on the hydrogen bonds. Let's say this is the DNA double helix. It won't come and cut here. Here, who cuts here between the hydrogen bonds? Which enzyme? Let's see if you know. Who's in the class? Sad. Restriction. Restriction enzyme, where do they cut? They cut here. They cut like this. Wait. Let's say this is the double helix. Okay, these are the hydrogen bonds. If the restriction enzyme came to cut, will it cut like this between here, or will it cut the hydrogen bonds like this? Will it cut horizontally or vertically? In the DNA, we have two kinds of bonds. No. In the DNA, we have two kinds of bonds, okay? We have the hydrogen bonds and we have the phosphodiester bonds. Let me try to draw, or let me try to write a sequence of bases, of nitrogenous bases, to see how does it cut. Let's say we have A, C, T, C, G, A, and we have here T, G, A, G, C, T, T. Okay, between them here we have two hydrogen bonds, 3, 2, 3, 3, 2, 2. Between these here, these are called the phosphodiester bonds, between these here. This is the hydrogen, and the one up is called the phosphodiester. Restriction enzymes, okay? Restriction enzymes, they cut the phosphodiester, they cut here, on these ones. However, the helicase enzyme, it cuts the hydrogen, it cuts here. Okay. Let me delete. Okay. Perfect. Now, so the multiple cloning sites, multiple cloning, yeah. So it's a region containing several restriction enzymes. So it has restriction enzymes. Okay. Sites where foreign DNA can be inserted. So the vector components are the origin of replication, are the selectable or the antibiotic resistant genes, and the multiple cloning sites where the restriction enzymes exist when the foreign DNA is inserted for the cloning to happen. The last component of the vectors are the promoters. Promoters are a sequence of the gene of interest that controls the gene expression. Okay. Okay. Before we continue, I want to speak about the penicillin because it was one time asked in the Imat. So the penicillin, it's a group of antibiotics okay, originally derived from Penicillium fungi discovered by Alexander Fleming. By the way, in Italy, specifically in Parma, we have a whole street which is called Via Alexander Fleming because he's the one who discovered this. You know, like Italians usually, when one of them discover something, they name a street in his name. Just so weird here. So discovered by Alexander Fleming. In 1928, it was the first antibiotic to be used widely, revolutionizing the treatment of bacterial infections. Penicillin is a kind of antibiotic that protects the bacteria from any viral infection, from bacteriophages or whatever. I mean, that protects the virus from bacterial infections. Okay? How does this penicillin work? So, it works by interfering with the synthesis of bacterial cell walls specifically targeting the enzyme which is called the transpeptidase. Okay, this enzyme is essential for the cross linking of the peptidoglycan. What is the peptidoglycan? If anyone of you remember, peptidoglycan, it's the substance that is covering the cell wall of the bacteria. Layer without. So what is the function of penicillin? It works by interfering with the synthesis of bacterial cell walls, specifically targeting the enzyme transpeptidase. What is this enzyme transpeptidase? Transpeptidase is essential for the cross-linking or the connection of the peptidoglycans or the peptidoglycan layer in this bacterial cell walls. If this penicillin came and attack this cell wall, attack this enzyme won't do its role, its essential role in this layer of the bacterium. If the bacteria don't have a strong cell wall, the bacteria will be destructed. No more bacteria. So it tries this penicillin. It's a kind of antibiotic that tries to kill the enzyme or tries to attack the enzyme, which helps in the connection and the production of the important layer of bacterial cell. And by this, the bacteria will be killed indirectly by the penicillin. Okay. We have different kinds of penicillin. We have penicillin G, we have penicillin V, we have amoxicillin, and others. Okay. Penicillin G is also known as benzyl penicillin. Penicillin V is known as phenoxymethyl penicillin. Okay. Amoxicillin is known as amoxicillin and others. These different types, you should not know each one of them, what's the main function of them. They have slightly different characteristics that differ them, but all of them, they try to destruct the peptidoglycan cell wall of the bacteria. So they all have the same function, maybe slightly different from the way they try to destruct it. However, it's the same result. Okay. Now we finished about penicillin. Let's continue speaking about where were we. So we were speaking about, let me see. Perfect, yeah. Now let's speak about cloning. Okay. What is cloning? So cloning is the process of creating identical copies of a DNA sequence, cell, or even organism. In molecular cloning, the goal is to insert a DNA fragment often a gene, into a vector so that it can be replicated or expressed, of course, in a host cell. Okay? So what is cloning? It's a process of creating identical copies of a DNA sequence, cell, or even organism. In molecular cloning, the goal is to insert a DNA fragment, usually a gene, into a vector so that can be replicated or expressed in a host cell. So let's say we have here a plasmid vector. We insert a DNA fragment inside this vector, and then it will become, let's say that the DNA fragment is like this. Okay, and this is the vector. The vector will end up looking, after cloning, it will end up looking like this. Yeah. Let's see how the molecular cloning is done. Okay, let's see the steps of molecular cloning. So, first step of this cloning is isolation of DNA. Second is digestion with restriction enzyme. Third is the ligation. Fourth is the transformation. Selection and screening. Replication and expression. Okay, so how does this molecular cloning happen? It happens on 6 steps. The first step is to isolate the DNA. The DNA of interest is isolated from the organism. We take out this DNA out of the organism, we isolate it. Second, we get restriction enzymes because restriction enzymes, they cut the DNA on a specific sequence, either from 5' to 3' or from 3' to 5'. It depends if the DNA is coding or non-coding. The DNA and the vector are cut with specific restriction enzymes to create compatible ends. Number 3, we add the ligase enzyme, ligation. This DNA fragment is inserted into the vector using the enzyme DNA ligase, which seals the DNA into the vector. DNA ligase helps in merging the DNA of the DNA fragment of the organism inside the DNA of the vector, which is the vector. So it tries to merge them. The main enzyme in cloning is the ligase enzyme. This is the most important enzyme you should know. So the ligase helps merging the DNA with the vector. Transformation, the recombinant vector. What is the recombinant vector? Is the vector that has both the vector and the DNA, which is out from the organism by restriction enzyme. This recombinant vector is introduced into a host cell. Who is the host cell usually in cloning? Host cells usually in cloning are bacteria. Let's take an example, because anyways, no prokaryote can be able to replicate without a body. The prokaryotes cannot replicate outside, only for some exceptions. However, most prokaryotes are so hard to replicate without a host cell, without a body inside, like they be inside and replicate on the side of this body. So they need a kind of bacteria. They need a host cell to be able to transform, to be able to replicate and express this recombinant vector, which we ended up from the vector and the DNA together. The transformation here happens. So the recombinant vector is introduced into host cell. Let's take it to example, Decoribacterium, via transformation. Transformation is the name of the process. This can be achieved by heat shock, electroporation, or other methods. Selection and screening. Cells that have taken up the vector are selected using the selectable marker. We spoke about the selectable markers, which are the antibiotics. Screening methods like blue-white screening are used to identify cells that contain the recombinant DNA. So it's a kind of screening that the recombinant ones are colored. However, the ones which are not recombinant, the ones who are only vectors or who are only DNA restricted DNA, they won't be selected. The only ones which can be selected are the ones which are colored, which are the recombinant DNA. Because if this recombinant DNA is not chosen and any kind of DNA other than this recombinant is chosen, the ending up organism won't be a cloned organism, he will be a normal organism, and all with what we did won't make sense. So we need at least a process, which is the screening process, to help us identify which from these DNA is the recombinant one and which of them which is not recombinant with another vector. Replication and expression, the host cells replicate the recombinant vector, producing copies of the DNA of interest. If the vector has a promoter, the gene can also be expressed in the host cells. So replication expression, they entered the host cell, this recombinant vectors entered them, lived in the host cell, they replicated, they are expressed. This host cell have a promoter. This promoter helps in this replication and expression. Okay? So if this host cell have a promoter inside, this gene of interest will be expressed inside this host cell. Okay, if it does not have a promoter, because promoter helps in gene expression. However, the repressor helps in gene repression. Okay, so if you want, you can take it as a note. Promoter helps in gene expression. Repressor helps in gene repression. Means does not allow the gene to be expressed. Okay. Now let's speak about the PCR. Or the polymerase chain reaction.
Sorry,
So,
can you repeat promoter helps and what?
okay. So, promoter helps in gene expression. Repressor helps in gene repression.
Thank you so much.
You're welcome. Okay. So, before we continue, I remembered something. Yeah. So, Okay, before we continue about PCR, I want to speak about something else, which is also important for the Imat. So, we will speak about the acetylation and methylation and their effects on gene expression. What is acetylation? What is methylation? Acetylation is the addition of an acetyl group. What is the acetyl group? The acetyl group is the CH3CO. Okay. To a molecule. So when we have a molecule and we add this acetyl group, this functional group, this molecule will be acetylated. Okay. In the context of gene regulation, it typically involves the acetylation of lysine residues on histone proteins. What are the histone proteins? Are the proteins at the end of the chromosome. Only in eukaryotes, okay? Histone proteins are the proteins which are on the end of chromosomes only in eukaryotes. What is associated with histones on the DNA, on the chromosomes? Telomeres are associated with, let's speak about this before we continue. So let's say we have this chromosome. At the end of the chromosome, we have proteins which are called the histones protein. These histones, we spoke about them, I guess, in one of our sessions. So they coordinate with cyclins to allow the cell cycle to continue. Okay, this we spoke about it. We spoke about all the types of histones, all the types of cyclins, everything. However, associatedly with the histones, we have the telomeres. Telomeres are here at the end of the chromosomes. Why does these telomeres exist? Telomeres, so they are associated part of the DNA at the end of the DNA, because when the cell cycle happens, when so much cutting of this chromosomes is happening, the chromosome will start to be smaller. And when the chromosome is getting smaller, then the chromosome will disappear at the end of so many mitosis and so many meiosis. If the chromosome disappear, our genetic material will disappear. We ourselves, we won't have characteristics. So that's why telomeres exist. They help in the elongation of this genetic material, in the elongation of this chromosome. Okay? However, if this telomere is cutted and then another telomere is made again, is produced, and then it will be cut during this cell cycle, and then another telomere will be there. Who will allow this? Who will help this association of telomeres? It's called the telomerase enzyme. So it helps in this synthesizing and in this continuous existence of telomeres at the end of the chromosomes. Okay. Now we were speaking about acetylation. So acetylation, when we add a CH3CO group for the molecule. In gene regulation, it typically involves the acetylation of lysine. Lysine, it's an amino acid residues So the acetylation is when we add this acetyl group, or we make the acetylation of lysine residues on the histone proteins by the enzymes which are called the histone acetyl transferase HATs. Okay. How does this acetylation affect the gene expression? Histones are proteins around which DNA is wrapped to form chromatin. When histones are acetylated, this molecule of this protein of histone have this group inside it. Okay. The positive charge on lysine residues is neutralized. Lysine is usually positively charged amino acid. Okay. So this positive charge, which is on lysine, okay, will be zero, will become zero. Okay. So this histone proteins were normal. Okay, it's at the end of this chromosome to make the chromatins and helping in the cell cycle, whatever. Acetylation happened immediately. When acetylation happened, this lysine will be acetylized and then acetylated, I mean. And then what happens? This histones will be acetylated. I'm sorry. The histones will be acetylated means we will add this acetyl group to the histones. The moment that this acetyl group is added to the histones, this lysine, which have a positive charge, will become neutral, will become zero charge. If this lysine becomes zero charged, it reduces the interaction between histones and the negatively charged DNA. Okay, so if this lysine is zero now, there won't be an interaction between the histones and the DNA again. If the DNA and the histones does not have an interaction between them, a negative charged interaction between them, the chromatin structure will be more relaxed. There is no huge interaction. There is no strong interaction between the histones and the chromatin. It will end up with a relaxed chromatin structure, which is called the euchromatin. Okay. So this acetylation, this relaxed chromatin, will allow transcription factors and RNA polymerase easier to access to DNA. Why? Because the euchromatin is normally super simple kind of chromatin, which has no associated things together, or it's not really complex. So when it's not really complex, the transcription factors and the RNA polymerases, they will be able to super easily access to the DNA. If it was really easy to access to the DNA, the gene transcription will be facilitated, will be easier. So after this gene transcription is facilitated, gene activation is easier. So acetylation of histones is generally associated with gene activation or high gene expression. So acetylation, it increases the gene expression. How does it increase the gene expression? We're gonna add this acetyl group on the histone proteins at the end of the chromatin. The moment it's added, we have two charges. We have the positively charged and the negative charge. The positive charge comes from the amino acid, which is lysine, and the negative charge comes from the ends of the DNA and the histones, of the chromatin and the histones. The moment that this acetyl group is added to the histones, the lysine will be zero charged. When it's zero charged, now there is no attraction because usually positive attract negative. However, zero does not attract no one. So when it is zero charged, we don't have an attraction between the lysine and between the histones and the chromatin. When the histones and the chromatin has no huge interaction between them, what will happen? A very simple kind of chromatin will end up, which is called the euchromatin. When the euchromatin is simple and facilitated, it will make it really easy for the expression to be activated. When this expression is activated, what happens? It will increase the gene expression. So acetylation of histone proteins, they increase the gene expression. Okay? Let's speak now about the methylation. So, however, if we remove this acetyl, let's say the histones already have acetyl, okay? And they told you in the Imat, so we removed an acetyl group from the histone. What will happen? Low gene expression. So, let's say they told you we have a histone. We added acetyl group. What will happen here? High gene expression. However, if they said we have histone, okay, already have an acetyl group, and then we broke it down and we took out this acetyl group, this will decrease the gene expression. Okay, so if it came opposite, you should know like how to do it. Now let's speak about the methylation process. Okay. Okay, so methylation, it's the addition of methyl group. What is methyl group? CH3. Okay. So in gene expression, if we added a methyl group, means a CH3, to cytosine bases, okay, in DNA, or to a certain amino acid on histone protein, what will happen to gene expression? Let's speak about it. So, cytosine bases, which is the C base, have guanine, cytosine, adenine, whatever, thymine. So let's speak about the cytosine base. If we added a methyl group to this cytosine bases, okay, in the context of CpG, dinucleotides, CpG is a cytosine followed by guanine in the DNA sequence. So means like CG, CG, CG in this way. Okay, this is called the CpG. So this sequence is catalyzed by enzymes called the DNA methyltransferase. Okay, or DNMTs. So what happens if we added a methyl group to this cytosine? If we add here a methyl group on each cytosine and the CpG dinucleotides. So if we have a DNA base, okay, a DNA sequence, and this DNA sequence just have a C and G, have nothing else, okay? CG, CG, CG. This is called DNA CpG dinucleotide. Why di? Because we just have two kinds of bases. So CG, 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 G, C, C, G, like this. On each C here, we added a methyl group. On each cytosine, we're adding a methyl group. Okay? What happens? We start from the promoter region. Promoter region is the origin, is the beginning, is the origin. So we go to the promoter region of the gene. Usually, the promoter region is associated with gene repression. Why? Of course, if it was because we started with C and C is methylated, we added the methyl group to the C. So this promoter region will be binded or associated with gene repression. Why? Because methylated DNA can recruit proteins that recognize methylated cytosines, leading to chromatin condensation and suppression of gene transcription. Okay, And so this methylation to this cytosine on the promoter of the gene will lead us to a condensed chromatin. If the chromatin is condensed, means it's super complex. When the chromatin is complex, or also called the heterochromatin, when it's really complex, this gene expression will decrease, so low gene expression. So the more we methylate the cytosine, the more gene expression will be reduced. The more we acetylate histone, the higher the gene expression will be. Okay? This reminds us of a topic which is called the inactivation of X chromosome. Okay? Let's speak about the inactivation of X chromosome. Okay? Let's speak about this inactivation. It was also asked in the talk 7 times in the PDFs that were for this year. So, as we said, acetylation, high gene expression, methylation, low gene expression. Let's speak about now the inactivation of X chromosome. It's a vital process in mammals that balances gene dosage between males and females. It's an important process. Since females have 2 X chromosomes and males have only 1 X chromosome, XCI ensures that females, like males, have 1 functional X chromosome per cell. So the X chromosome activation ensures us that since although the female has 2 X chromosomes and the male has XY, there is a balance between both females and males. So it says, this topic says that one of these X's in the female is active, however the other is inactive. And says that the X which is in the male is active. So now we have a balance, 1 active here, 1 active here. You know? So this is a kind of balance between the X chromosomes in nature. So mechanism of X chromosome inactivation. Let's speak about the mechanism. So we have the random inactivation and the non-random inactivation. Okay, so today we're gonna study until 5, from 1.5 until 5. Okay. So we have the random inactivation. In placental mammals, XCI is random on most cells. This means that either the maternal or the paternal X chromosome can be inactivated in a given cell. So it's not important that just the mother have the inactive X, it can be also the father, okay? This randomness leads to the mosaic pattern of X chromosome activity across different cells. So here can be however, the non-random inactivation, there in some species or specific cases, there can be a preferential inactivation of one X chromosome over the other, but this is less common. So it can be either maternal or paternal. This is the most common. However, the non-random is the less common. So inactivation of X chromosome is mostly random process. So how does this mechanism happen? Okay, so listen to me. Cells have a mechanism to count the number of X chromosomes. This involves the detection of the cis RNA, a long non-coding RNA that is crucial for initiating XCI. In females, we have two X chromosomes. One of the X chromosome will become coated with the cis RNA. What is the cis RNA? Long non-coding RNA, which is important for the XCI or an activation of X chromosome. So what happens if we go to the maternal part? Maternal means it has to access one of the X chromosome will become coated with this cis RNA, marking it for inactivation. It will be colored, it will be coated, it will be associated with this cis RNA. It will come, this non-coding RNA, it will come and sit on this X chromosome of the mother, one of them, not two of them, only one of them. And then it will mark it, it will be like, okay, look, I'm here inactivated. So to mark it for inactivation. Okay. Perfect. So, second, what happens? Chromatin changes. The X chromosome that is inactivated undergoes significant changes in chromatin structure. Now, this X chromosome is inactivated because the cis RNA is associated. So, what happens here? This inactivation, okay, undergoes so many, the X chromosome which is inactivated will have so much changes in the chromatin structure. It might have addition of repressive marks, it might have DNA methylation, like something might happen, acylation, methylation, whatever, okay? And it will become to a very condensed chromosome, which is a chromatin, which is called the heterochromatin. Usually euchromatin is the simple one, which ends up from acetylation. However, methylation ends up with the condensed chromatin, which is called the heterochromatin. When this X chromosome is inactivated, the chromatin will become condensed. It will change in structure from euchromatin to heterochromatin because of the excess RNA binding. What is the bar body? The bar body is one of the most important things you should know about this topic. So, the bar body this inactivated X chromosome, it won't be as an image of a chromosome anymore. It won't be as an image of something called a chromosome. It will be an image of something called the bar body in the nucleus. So you will be seeing a bar body in the nucleus. This is exactly the same inactivated X chromosome, which is a dense, highly compacted structure. It's a kind of chromosome which is condensed, which is the heterochromatin. Good afternoon, Saeed. Yes, the class is recorded and I will upload it today, okay? Don't worry. Okay, just 1 second. Okay. So we were speaking, Saeed, about the inactivation of X chromosome. Okay, let me try to summarize for you. Today our class is until 5. We have so much to do. So we were speaking about the inactivation of X chromosome. We said that this X chromosome, usually males and females, both of them have X chromosome. But in males, we have more than one. We have two, and in males, we have one, and in females, we have two. So a kind of balance we have between the X's. The X of the female, one of them will be activated, the other is inactivated. However, the one in the male, it can also be activated or non-activated according to the random inactivation process. What happens in this inactivation process? We have a kind of RNA, which is non-coding RNA. It's called the cis RNA. This kind of RNA will come and bind, not it won't bind, but it kind will it will kind of associate the X chromosome, one of the X chromosomes of the female. And then when it associates, what will happen? This X chromosome will be inactivated. When this X chromosome is inactivated, what will happen? The chromatin of the chromosome will become super condensed. It won't be anymore a euchromatin, it will become a heterochromatin. This heterochromatin will end up being condensed continuously. It will condense, be super strong, super thick, and this will end up with a bar body. So what is the bar body? The bar body is the inactivated X chromosome in the nucleus, which is dense, highly compacted structure. Okay. Now we have something after this inactivation. Let's say that we have that one of the X chromosome is now inactivated. It went and it sat in the nucleus as a shape of the bar body. Perfect, perfect. This inactivation, it is stably maintained through cell divisions. If meiosis happens, since this is XY means sex cells. So if meiosis happens, what will happen? It means that the same X chromosome, which is inactivated, it will remain inactivated in all of the daughter cells. Okay? Why? Because it's trying to preserve the balance of X-linked gene expression throughout the generation. So you should know that X chromosome, which is inactivated, will go and sit in the nucleus as a very compacted structure of chromosome, which is called the bar body. After the inactivation of this X chromosome, let's say this cell will undergo meiosis, it will undergo any cell division. These cell divisions that happen will end up with daughter cells which have at the end also one inactivated X chromosome. This is called the maintenance character of the inactivation of X chromosome process. Yeah, perfect. So for inactivation, we're done. Okay. So let me see, what did we speak about? Yeah, now we have to start with the PCR. Polymerase chain reaction. What is the PCR? PCR is a powerful technique used to amplify. One of the most important things is amplify DNA sequence, generates millions to billions of copies of a particular DNA segment and this technique helps in so much other techniques like cloning. So it helps in cloning, it It helps in gene analysis, in forensic science, and any other things. So the main reason of why do we do the PCR is the amplification of DNA sequence. Okay. Okay, so the components of PCR. We have the template DNA, which is the DNA that we're taking to amplify. The primers. Okay, the Taq polymerase, or the polymerases, let's say. Nucleotides. Buffer solution. So these are the components of PCR. The template DNA is usually the DNA sequence that we want to amplify. Okay. Okay, so we said that. Yeah, so the DNA sequence that we want to amplify, the primers are short sequences of nucleotides, or also called the oligonucleotides, which are the small nucleotides, short sequences of nucleotides. So the targets DNA start and endpoints. They provide a starting point for DNA synthesis. Polymerases, like the Taq polymerase, which is usually and mainly used, these polymerases is a heat stable, a thermostable. It's called the thermostable polymerase. A heat stable DNA polymerase derived from Thermus aquaticus. This enzyme, or this polymerase, synthesizes the new strand by adding nucleotides to the primers. Okay, so it adds nucleotides to the primers. Now, of course, we want the nucleotides, the DNTPs. The nucleotides are the building blocks. What are the nucleotides? Are the adenine, thymine, cytosine, guanine. These are used to synthesize the new DNA strand after the PCR, and the buffer solution maintains the pH, as we said. So pH maintenance. And ionic conditions for the activity of DNA polymerase. Okay, so guys, what is PCR? PCR, it's a kind of technique that's used to amplify a DNA sequence. To amplify this DNA sequence, we need tools. The tools are the template DNA, which is used to amplify. It's the kind of DNA which is cut with the restriction enzyme that we use it to amplify the sequence. The primers, or the oligonucleotides, the polymerases like the Taq polymerase, which are thermostable polymerases, which are able to hold and stable in front of heat and high temperatures, derived from Thermus aquaticus. The nucleotides are these the building blocks used to synthesize the new strand, and the buffer solution for the pH maintenance in the PCR. Okay. So like the nucleotides, we add the nucleotides to the primers. When these nucleotides are added to the primers, this DNA will be amplified. Now let's see how does it happen. Let me draw the sequence. How does this happen? So steps of PCR. One is denaturation. Two is annealing, deformerizing. Three is extension or elongation. Four is repetition. What is denaturation? We have a double-stranded DNA, okay? We bring restriction enzymes, and we cut the specific part that we need to work on. Okay, now restriction enzymes, we cut it, we took this part. Okay, let's take the small part, double-stranded DNA. It will look like this. Go from 3' to 5'. Here we have 5' to 3'. So what happens? So we want to amplify it. What do? Step 1, we heat it. Step 2, we add the primer to cool it down because it's heated. So we add the primer. Okay, so we add the primer using the primase enzyme. So the enzyme here, which is important, is the primase. However, in cloning, it's the ligase. After we cool it, what do? Okay, so ions. We add ions, Mg2+, Na+. Okay, these are the template primers. Okay, and here we add the Taq polymerase. Which is thermostable. And then we increase the heat. We heat it much more. Why did we heat it usually after we add Taq polymerase? Because usually this polymerase is thermostable. It can hold heat. So if we heat it so much, this polymerase is able to hold it, right? And then here we repeat the cycle. We repeat the first and second cycle, which is first we heat, and then we add the primer, and then we still heat and add the primer, heat and add the primer until this DNA part is already amplified. Okay, let's speak more about the process. How does it happen? Each step of them. So denaturation. The double-stranded DNA is heated around 94, this to memorize, 94 to 98 degrees Celsius to break down the hydrogen bonds between the bases, resulting in two single strands of DNA. So this double-stranded, we heat it to break down the hydrogen bonds. This double strand, here we have hydrogen bonds. To break these down and to end up with single strand and single strand. So denaturation happens from 94 to 98 degrees Celsius, this range, and we are breaking the hydrogen bonds. Okay. Second step is the annealing. The annealing step, the reaction temperature is lowered to 50 to 65 degrees Celsius. So from 50 to 65 degrees Celsius. We lowered, we cooled down. Since we said here, we have the primers to cool down this reaction. Because the primers can only bind on a temperature which is cool temperature. However, the primers cannot bind on a high temperature. So what happens? We have to cool it down to allow the primers to bind. What does anneal mean? Anneal means binding. Okay. To their complementary sequences on the single-stranded DNA. We usually amplify a single-stranded DNA and not a double-stranded DNA. In the elongation, the temperature is then raised from the 65, it will be raised to 72 degrees Celsius. Why it's raised to 72? Because this 72 degrees Celsius, it's the optimal temperature of who? Of the Taq polymerase. So that's why we increased it, because Taq polymerase is thermostable, means it needs a high temperature to function. So we have to increase it to the optimal, to the much minimum temperature the Taq polymerase is able to function. The minimum temperature that this Taq polymerase is able to function is the 72. And here we start adding nucleotides, where we add the nucleotides to, so add nucleotides to the 3' end of each primer, synthesizing the new DNA strand. So Taq polymerase, what does it do? What's its function? It helps in adding nucleotides to the primers. So it helps adding nucleotides to primers. And then the cycle of denaturation, annealing, and elongation is repeated how many times? Cycle is repeated 20 to 40 times. Okay, doubling the DNA quantity with each cycle. So it tries to double the DNA quantity with each cycle. After 30 cycles, over 1 billion copies of the target DNA can be generated. So let's write this as a note. So the cycle (20 to 40 times), doubling DNA quantity with each cycle. After 30 cycles, over 1 billion copies of the target DNA can be generated. So here we amplified, or we doubled, we copied this DNA sequence, the single DNA sequence, because we use the restriction enzymes to cut this DNA helix, and we doubled already the DNA quantity. Okay. Now let's speak about PCR in cloning. PCR is often used, so PCR is often used to amplify the DNA sequence of interest before it is inserted into a vector of cloning. As we said, in the cloning, we have a vector, and we try to insert the DNA, the amplified DNA inside this vector. So this DNA that we, if you remember, we said that we have a vector, let's say plasmid, and we insert the DNA inside it using the enzyme that we spoke about. So to amplify, to do this, to be able to do this, no. So to be able to do this, this DNA, which is the DNA of interest, this one, must be amplified before the insertion of the vector. Okay. So so how does PCR enter cloning? The DNA must be amplified, means undergo PCR, and then cloning must happen. Why? Because if you have the if you have this, let's say that we have this DNA, okay, and it's double-stranded, okay? So when the DNA is double-stranded, to add another kind of DNA to it, it will be so hard to add because it's double-stranded. However, if it was single, another one will be single and bind to it. And if it was here also another single, this single will bind to it. However, if these two are, if it was double helix, it will be so strong to cut it from here by the enzyme. The enzyme won't be able to cut it because it's really strong, because it's double helix. We have strong, we have hydrogen bonds between them. So for this reason, this enzyme won't be able to cut it, and this vector, this cloning won't be able to happen. However, if it was cut by restriction enzyme, single by single, this vector will be able um, to merge his DNA with this DNA sequence. Okay, now let's speak about. So we're done with biotechnology, I guess. Okay. Before we continue, I still want to speak something. Okay. Okay, before we continue, I want to speak something also about biotechnology. Um, no, about inheritance. So sometimes we don't have mutations, but problem might happen in segregation of chromosomes. Okay, let's say this mitosis is happening. So what will happen, instead of having 2 here and 2 here, it will end up having 2 here and 0 here. Or it will end up having 4 here, and here it won't have nothing. It will be 0. So like this wrong segregation that happens during the cell cycle is called, this is called the random segregation or non-disjunction. So non-disjunction or ploidy or random segregation. Okay. What are kinds of non-disjunctions? We have the XO, which is called, we're speaking about sex chromosomes, Turner syndrome. We have XXY, Klinefelter syndrome. And we have XXX on 21 or on 18. It's called the trisomy 21 or trisomy 18. And let's say here we want to write the formula, genetic formula. We will say 47. Why 47? Because we have an additional chromosome here. Instead of 46, there are 47. And then we write plus 21. Okay. So here we write 45XO. Here we write 47XXY. Okay, this is the formula. There's something also called the what was it called? Yeah, the translocation. We speak about the translocation. Translocation from its name, it says it's speaking about the location of this gene. Let's say we have the karyotype. We all know what that's the karyotype. Karyotype helps in knowing if the person have any disease, if the person has any mutation, if the person is a boy, if the person is a girl, if the person has any hereditary disease, if the person is healthy, if the person is a female. So this karyotype, it's the arrangement of chromosomes on a screen. Okay. Let's say we have here this part is lost. So this chromosome, instead of being big like the other, the same size, two chromosomes must be the same size. Only the XX will be like this and the XY will be like this. Y is usually smaller than X, okay? And the sex chromosomes which are here usually. Like if it's a female, it will show like this. If it's a male, it will show like this. Okay? If he is a Klinefelter, it will show XXY like this. This in the karyotype. So Y is usually smaller than X. Now let's say that we have here on chromosome 21, we have a part of this chromosome is taken. So it will look like this on 21. 21 must not look like this because it does not have a Y. It's not a sex chromosome. And let's say that chromosome 1 looks like this. So this part traveled from 21 and came and sat on chromosome 1. It won't disappear, it will go and sit in a place. But it traveled from 21 and sat on 1. At the end, this person will end up the same person, but it's only the karyotype which is changing. His genotype will stay the same, his phenotype will stay the same, nothing will change, no genotype, no phenotype. Genotype and phenotype are constant. After translocation, it won't change nothing. It will just change the karyotype. Yeah, you should know about the gel electrophoresis. It's so easy. I will say it now. So you have a genotype and karyotype will stay if genotype and phenotype will stay constant. However, the karyotype will change, and the part of this chromosome which is sitting on 21 will go and sit on the one, and ending up with a something called translocation. But also the genetic formula will stay 46 XX or 46 XY. It will stay the same, nothing else. Okay. It won't change nothing. Even the formula will stay the same. Okay, so perfect. So you were speaking about gel electrophoresis. Gel electrophoresis is a technique to separate the DNA, the RNA, the proteins, depending on their size. Okay, depending on their size, depending on how are they, on their structure, on their charge. So how does this electrophoresis happen? First, they have to prepare in the lab. In the lab, they prepare a gel. This gel is made up usually of some material which is called the agarose gel. Agarose gel are used for nucleic acid fragments, and another kind of gels are called the polyacrylamide gel. This for big fragments like DNA, nucleic acids, and the polyacrylamide gel is for small fragments like protein. Okay, this is called the gel electrophoresis. Okay. So what happens? This gel is poured into a mold, and then it's allowed to become solid. It will form a matrix with microscopic pores. So after we pour it here, Here we pour the gel, one of them. If it's a nucleic acid, we pour agarose gel. If it's a protein, we pour polyacrylamide gel. Okay. After we pour this gel, we will leave it some time. It will become a kind of matrix, and then this matrix will have pores inside. Here we have to prepare the sample. Samples are mixed with a loading buffer that contains dyes and glycerol to help visualize and to see for the sample to start to give a color or to give a like a phosphor color, just to a kind of dyeing, just to be able to know, oh, this nucleotide or this part of this nucleic acid is the nucleic acid which should undergo this, which undergone this electrophoresis. So what happens in the electrophoresis? Since we said electrophoresis, this means that we're using electricity. So here we have to apply voltage. This gel is placed in an electrophoresis chamber. And this chamber is filled with a buffer solution. An electric current is applied across the gel. We put here a electrical what was the name of this? Node? I forgot. So I put this here, which allows the current to be applied inside this gel. And when this current is applied, what will happen here? Charge will be applied. And so much molecules will start to, charged molecules like ions will start migrating throughout the gel. DNA, RNA, or protein molecules move through the gel towards the positive electrode. Yeah, this is called electrode. So it will move towards the positive electrode. The DNA, the RNA, and the proteins will start moving towards the positive part of the gel. Okay? Because to the positive electrode, because they are negatively charged usually. DNA, RNA, and proteins are usually negatively charged. So they will, because positive electrodes attract the negatively charged, so they will go and be attracted to them. Why are they negatively charged? Because DNA has a phosphate backbone. And RNA has also a phosphate backbone, and phosphate is usually super electronegative, has very high electronegativity, especially the phosphate backbone is associated with, because it's a phosphate backbone and it's super electronegative. However, for the proteins, why they are negatively charged? Because they have a net negative charge, because they have usually the carboxylic acid part, which has the two oxygens, which are really electronegative. So that's why they are attracted to the positive part of the electrode. So now the separation, how does it happen? Smaller molecules move through the gel matrix more easily and travel further than larger molecules. So smaller molecules, okay, are faster and go further. Why? However, big are the opposite. Let's imagine we have a person who is skinny and a person who is fat, and both of them are running. Of course, the skinny will reach the end much faster than the fat one, because the fat one has to hold his size and walk. However, the skinny one don't even have a size to be, or have a good weight to be holding himself. He will just go running, so he will reach faster. Same thing happens here. If we have a small molecule, it will reach much faster and much further to the positively charged and to reach before the big molecules. Okay. How do we visualize it? How do we know this happens in electrophoresis? Since we said that we already died, so we gave a color, so we're able to know which one of them reached before or after. After the dye, they will be colored, and after they are colored, this nucleic acids and these proteins, we will know which part or which one of these molecules reached faster, reached further, because they will look like this at the end. And you will see here this colored, this colored, this colored, and we will see who reached first, who reached last, you know. Why do we do electrophoresis? It helps in determining the size of DNA fragments, in cloning, in sequencing, and even genotyping and PCR. Um, they even help in gene expression and RNA integrity. And this for the DNA and RNA. However, for the protein, why do we use electrophoresis for protein? Using the polyacrylamide gel. So protein separation, they separate proteins by size or charge for further analysis and to detect specific proteins using staining methods or immunoassays. So using this staining or the dyeing methods, we're able to know what kind of protein is this. If it reached before, this means that this protein is highly negative charged. If this protein is highly negative charged, it's most likely that this protein um, has called monomers, which are the acids monomers, amino acids, which are super acidic amino acids and super negatively charged amino acids, other than the non-charged ones or the or depending on the polarity or the non-polarity. Let's say that if we have two kinds of proteins, let's say we have one protein which has amino acid which is the lysine, and another protein which the amino acid is glutamic acid. Of course, the glutamic acid will reach the positively charged rod much before than the lysine, because the lysine is usually positively charged. However, the glutamic acid is super negatively charged amino acid. And in this way, according to the colored one, we're able to know which one of them is lysine and which one of them is the glutamic acid. The one which reached the first is the glutamic acid, the one which reached later is the lysine. Okay, perfect. This about electrophoresis. I don't think you should know more. So just helps in the size and detection, this thing. Okay. So for electrophoresis, we're done. Yeah, I wanted to speak also about something. Okay. So okay. Let's speak about gene cloning. No, we spoke about cloning. We spoke about that the host cell is the bacterial cell. Okay. Yeah, this one. Okay. Let's speak about the gene cloning via reverse transcriptase. So what happens here? We want to do gene cloning via reverse transcriptase. So of course, in cloning, what is the main enzyme that we use? The main enzyme is the ligase enzyme. So let's say we have this two single-stranded DNA. Let's say we have here CAATTC. Let's say we have CTTAAG. And then we bring the restriction enzyme and we cut them. And then we will end up with this. Okay. Here we will have a sticky end. And then what happens here? The ligase comes and join them again. Here the ligase come and join these two again. So this gene cloning via reverse transcriptase. What does the reverse transcriptase do? We studied already that the DNA after transcription becomes RNA. RNA after translation becomes protein. What does DNA transcriptase do? It makes the RNA become DNA. This is the reverse transcriptase action. Instead of going from DNA to RNA, we go from RNA to DNA. Okay, and this mainly the reverse transcriptase enzyme mainly is associated with the HIV virus. So the moment you see HIV, put in your mind it contains reverse transcriptase. Okay. Another thing, PCR is the only biotechnology process that does not involve ligase enzyme. Ligase enzymes are only involved in cloning in all other vectors and whatever, but they are not involved in PCR. PCR just involve primers, polymerase, nucleotides, polymerases, and these things. However, the cloning have the ligase. Okay, perfect. Before we continue, I want to speak about something that so usually on the biomolecules that we have, let's say we have a nitrogen ring, okay? Usually on one prime means like the first, let's say 1, 2, 3, 4, 5, 6. Okay, so one prime, it usually holds an organic compound. Okay, or nitrogenous bases. 3 prime usually holds the OH group or the hydroxyl group. 5 prime usually holds the phosphate group. Okay, these also for memorizing. Another thing you should know, to study the karyotype that we already spoke about, the karyotype, Rafi. Which phase of mitosis do you think we should stop the cycle to be able to study the karyotype? Let's see if you will know it. Let's say I want to draw the karyotype. On which phase should I stop the cell cycle to be able to have this chromosomes and to be able to see them on a screen? Okay. So in metaphase, we stop, mitotic phase in metaphase, specifically why metaphase? Because in metaphase, the chromosomes are mostly condensed and We need them to be super condensed to be able to see them clearly on the screen. Okay, another thing. Okay. I want to speak about something in gene regulation of eukaryotes. So gene. We have two kinds of genes. We have the constitutive genes and we have the facultative genes. The constitutive genes are always expressed and vital for function. Are the main important genes, you know. However, the facultative genes are due to trigger. Okay. Now, I want to speak about the gene regulation in prokaryotes. So, prokaryotes like E. coli bacteria typically prefer glucose over lactose due to the efficiency of glucose metabolism. When glucose is not available, means no glucose, who activates in the prokaryotes? Like operon activates in prokaryotes, okay, to enable the use of lactose as an alternative carbon source for the prokaryote. Okay. Why does this lack operon activates? Why are they asking for carbon? Why do they want to use glucose? Of course, if there is no glucose, they have alternative, which is the lactose. Okay, this system, this regulatory system, allows the bacteria to adapt to change environmental conditions and optimize its energy production. Environmental plus energy conditions. Okay. However, the lack of pheron, like the thing, how it works and the mechanism, I don't think it's asked for the IMAT. But you should know that we have repression, we have promotion, we have it's normal as if we have operator, promoter, anything, induction. Let me see if it's asked for the IMAT. Give me 1 second to explain it. Okay, so okay, I'll try to explain it in a way, but like in a fast way, because I don't know to be honest. Okay, let me explain it in a fast way. So, have the lack of balance. So, we have the preferred carbon source that you already spoke about, which is glucose. E. coli and many other bacteria prefer glucose as their primary carbon source because it can be metabolized more efficiently to generate energy. Catabolite suppression. When glucose is present, it inhibits the use of other sugars through a mechanism called catabolite repression. If we have glucose in the body of the prokaryote, it won't allow the prokaryote to use any other kind of sugar. It just uses glucose. Why? Because it's mediated by the CAMP or the cyclic AMP and the catabolite activator protein, the CAP protein. High glucose levels lead to low CAMP levels. Why? Because high ATP levels, which in turn results in reduced binding of the CAP to lac promoter, then suppressing the expression of the lac operon. However, if we don't have glucose, this bacteria is not in a place, in an environment that contains glucose, what happens? It will take lactose as an alternative. When glucose is scarce but lactose is available, E. coli can switch to using lactose. This involves the lac operon, a set of genes responsible for the uptake and metabolism of lactose. Okay? So, lac operon components, we have genes in the lac operon. The lac operon includes three key genes. The lac Z, the lac Y, and the lac A. The lac Z encodes for the beta-galactosidase, which breaks down lactose into glucose and galactose. The lac Y encodes for the lactose permease, which facilitates the entry of lactose into the cell. This helps the entry of lactose. This cuts the lactose into glucose and galactose. And the lac A encodes for the thiogalactoside transacetylase. This detoxifies byproducts of lactose metabolism. We have the LAC1 encodes the lac repressor protein, which binds to the operator region and inhibits the transcription of the lac operon in the absence of lactose. We have the operator, the site where the repressor binds to block transcription. So the repressor binds to the operator. The promoter is the site where RNA polymerase binds. Why? Because it wants to initiate transcription. The induction. When lactose is present, it binds to the repressor protein, causing a conformational change that prevents the repressor from binding to the operator. If induction happens, how induction happens, lactose come and be present and don't allow the repressor to bind to the operator again. It will change the conformation, so they won't have receptors to be able to bind together. This allows RNA polymerase to access the promoter and initiate transcription of the lack of peron genes, enabling the bacterium to metabolize lactose. So usually the induction happens when there's no glucose. No glucose, the repressor won't bind to the operator. In this way, it will give it a sign, okay, we don't have glucose. Now, yalla, activate the lac proteins for me to be able to start the lactose metabolism. LacZ will open, beta-galactosidase will be there to start to break down all the stored glucose into glucose, all the stored lactose, or this lactose into glucose and galactose. And then like Y, it will produce more lactose by lactose permease, and like A will be taking out everything which is waste products or byproducts outside this octaron. Okay, done with this. Okay. Okay, now I want to speak about some enzymes before we continue. Okay, so we have the helicase enzyme and transcription. We have the gyrase enzyme. We have the topoisomerase enzyme. And we have the ligase enzyme. This is for PCR. This is for cloning, not PCR. Okay. Helicase, it unwinds DNA. يعني it separates the double-stranded DNA and makes them single-stranded. Gyrase, it relieves torsional strain by introducing negative supercoils, and it's specific to bacteria. Let's say we have here DNA, and here we have DNA, and here we have a helicase trying to unwind them. Okay, so let's say here helicase is unwinding them. This is helicase working and cutting, and then they will start being like this. And here the hydrogen bonds will look like this, like coils. Why they will look like coils? Because they are cutting. When they are cutting, they will look like this. When you cut a rope, it will look like this, right? So the gyrase will come and try to make them again look like this, straight. So it will try to get over this torsion, this supercoiling torsion, and help them to come back to normal. Okay. Topoisomerase. So topoisomerase, its enzyme that manage. Topoisomerase is the same as gyrase. Gyrase is a kind of topoisomerases. It's an example of topoisomerases. Gyrase and topoisomerase are the same. Ligase, we said it joins the DNA fragments by forming phosphodiester bonds, completing the DNA after replication or repair. So ligase, they form phosphodiester bonds. Let's say that this DNA, this double helix, is cut. It will become like this. Here it's cut. It will come bring them back together. Here we have the phosphodiester bonds between them up, and here the hydrogen bonds. It works on this horizontal and not on the vertical, the ligase. The helicase, it cuts, it unwinds the DNA, means it cuts the hydrogen bonds. From its name, helicase, hydrogen bonds. It makes it single-stranded. This means it cuts inside, not on the sides. The topoisomerases and gyrase, they try to manage the supercoiling that's happening after the cutting of this hydrogen bond. Okay. Okay, now I want to speak about something before we continue. I want to speak about DNA replication. DNA replication. So you should not know nothing about it. It won't be asked. The only part which will be asked is the elongation, the second part. So we have like 3 parts: initiation, elongation, termination. The only part you should know is the elongation for the Imat. Elongation, you should remember this. Okay. Here we have the mRNA. And you should remember that the mRNA will end up with a 5' cap. Okay. And you should know, okay, this is the RNA polymerase working here. And okay, and you should know that here we have that creates this 5' cap. No. Yeah. So what happens here? We start with the initiation. What will happen in the initiation? We will have a replication bubble. We will require ATP, and we will use the helicase, and it will cut it into 2 strands, 2 single-stranded molecules. After this single-stranded, what happens? Yeah. After the single-stranded molecules are there, they will look like this, and mRNA will start to appear. mRNA is appeared during this transcription. What will happen here? The mRNA will end up having a 5' cap. When it has a 5' cap, half of the mRNA will be outside and half of the mRNA will be inside as a result of the RNA polymerase action. Outside in the cytoplasm, we will have something called, not the cytoplasm, let's say the nucleoplasm, because the mRNA is inside the nucleoplasm now, it didn't go out. So in this nucleoplasm, we have something called the raw protein. This raw protein, it will cut the mRNA and will throw it in the cytoplasm. So it will cut mRNA and will throw it outside. So this part of the mRNA will be cut by the raw protein. Perfect? Perfect. So in the cytoplasm, we have something, a substance which is called the 7-methylguanine. This 7-methylguanine, it will protect mRNA from the enzymes in the cytoplasm. So from the enzyme. So mRNA will be made. The RNA as a function of, as a result of the RNA polymerase action, half of the mRNA will be outside the cytoplasm, will be cutted from by the raw protein and at its 5' cap. However, it will be protected from everything in the cytoplasm, enzymes and hydrolytic enzymes that might kill this mRNA by 7-methyl-guanine substance in the cytoplasm. After what happens, we will have a process which is called the spliceosome process. And of course, ending up with a bubble. After the transcription, we will have this bubble above. So we have the process which is called the spliceosome process. We will have the mRNA which was in the cytoplasm, having the 5' cap, which was protected by the methylguanine. So what happens here? We will have like this, 5' to 3' because it's an mRNA, so usually from 5' to 3' end. Here we will have the introns and here we will have the exons. Introns will form a bubble-like structure like this. And exons will stay from 5' to 3'. And then these bubbles will become a real bubble, like this super small between them, the distance. And they will be cut outside this. And then I will end up only having the exons. What are the exons? What are the introns? The introns are the non-coding part of the mRNA, the part that we won't use at all. However, the introns are the ones which we use for later to create the proteins from this mRNA. So these introns will be cut by a process called the spliceosome process. The termination, these bubbles will be there. In the termination, what happens? We will have the DNA polymerase helping. What will it help? This DNA polymerase and the ligase. So in the termination of this transcription, we have the termination process. We will have these bubbles. So ATP ligase will work. And the DNA polymerases will work. Let's say these dots are DNA polymerases, okay? So what happens? These DNA polymerases will stay inside to hold them all together to stay shaped in this way, to keep shape. Let's say one of these DNA polymerase is out, another bubble will form inside. We don't want these bubbles to be forming. We want it a stable shape inside. So DNA polymerases and ATP ligases will work, so this bubble shape will be out. Okay. One last thing. Let's speak about the DNA polymerase action. Okay, let's say we have, of course, we also just need the elongation, nothing else. We need the elongation and the termination. So let's speak about the elongation. So we have here from 5' to 3', and we have from 3' to 5'. This is the leading strand. This is the lagging strand. On the leading strand, we put Okazaki, this DNA polymerase will add Okazaki fragments, okay, only on one primase because the replication is continuous. So in leading strand, the replication is continuous. So only one Okazaki fragment, this DNA polymerase will add one Okazaki fragment on the primer. However, on the lagging strand, you will have so much Okazaki fragments, so much. Why? Each time one Okazaki fragment is created on the lagging strand, we will need a new primer. Okay? So who here works the most? Which enzyme works the most? Primase enzyme works the most. Because on the lagging strand, so much Okazaki fragments are added by the DNA polymerase. So each time one Okazaki fragment is formed, one primase is needed. And that's why primase works so much. On the leading strand, we only have one Okazaki fragment on one primer because replication is continuous. Okay, this also a note. You should have known. Perfect. If you want to speak about the pathway sequence, this is out of topic, but let's stay on it. So if you want to speak about the pathway sequence, from the kidney to the liver, we start with the renal vein. Usually we start with veins. So from the renal vein to the inferior vena cava. Why inferior? Because the liver is down. To pulmonary artery. Why pulmonary artery? Because usually pulmonary artery holds deoxygenated blood and vein hold deoxygenated blood. This is holding deoxygenated blood. This is holding deoxygenated blood, and this is holding deoxygenated blood. All of them are deoxygenated because they want to go to the liver. Liver helps in detoxification. Okay, so, and then it will go to the pulmonary vein. Pulmonary vein holds oxygenated blood. So this means it cleaned itself in the lung. And then it will go to the aorta. Aorta holds oxygenated blood. And then it will go to the, from the aorta, here now we have oxygenated blood. It will go to the hepatic artery. Hepatic artery holds oxygenated blood. So how the blood goes? They might ask you, what is the blood circulation from the kidney to the liver? If let's say we have blood going from the kidney to the liver, what happens? You should understand something, that all our body organs, they have deoxygenated blood. So when blood enters any organ, even the heart, when blood enters them, even the lungs, the blood will be deoxygenated. When blood leaves them, when blood enters them, it will be oxygenated. When blood leaves them, it will be deoxygenated. Okay, only the lungs, the blood enters them is deoxygenated, and the blood leaving them is oxygenated because he used the oxygen which is in our surroundings. Now let's speak about if the blood is going from the kidneys to the liver. Of course, if it's going out of the kidneys, means the blood leaving an organ. When the blood leaves the organ, this means it's deoxygenated. Which part of the kidneys holds deoxygenated blood? Of course, it's the veins. Veins usually hold deoxygenated blood. So renal veins are holding the deoxygenated blood. Vein usually go to the inferior vena cava. From the inferior vena cava, they will go to the pulmonary artery. Here we still deoxygenated blood. The pulmonary artery, of course, no blood which is deoxygenated will go to another organ of the body and stay deoxygenated. It must go first to the lungs and then go to another organ of the body, because it cannot be holding so much high CO2 and then go to the another organ and give the organ CO2. No, it must go to the heart, to the lungs, and then go back to another organ. So this means between the kidney and the liver, here we have an additional part which is the lungs. It will go to the pulmonary artery. Pulmonary artery is holding the oxygenated blood. It will take it to the lung. It will clean it and go with the pulmonary vein. Pulmonary vein is holding oxygenated blood. It will go to the aorta, and usually aorta takes oxygenated blood to all other organs in the body. So now here the organ that we have to go is the liver, and it will take it to the hepatic artery. Everything that have hepatic means cells. Hepatic cells are for the liver, means the liver. So it will take it to the hepatic artery, means the artery of the liver. However, the artery of the kidney is called the renal artery. Each word means something. Renal means kidney, hepatic means liver. Okay, pulmonary means lungs. So in this way, you can know them. Okay, this was also a question on the Imat. Now, let's speak about, spoke about so much things, I never knew that I have to speak about them. So, biotechnology, vectors, PTR, cloning done. Now, I'll speak about, let's be fast. Perfect. So, we spoke already about evolution, but we didn't finish it. So let me just be fast speaking about evolution before we continue. If we still had things to do, tomorrow we do another session. You don't worry. Before Monday, like the course will be done and everything. So because after an hour I have to go to work, so I guess we will do 1 more hour. We might still have, of course, we will still have like a 2 hour session. We do it tomorrow, okay? Because after tomorrow you'll start solving. So genetic variation. So the key concepts in evolution. We have genetic variation is the difference in DNA among individuals within a population. Okay, it provides the raw material for evolution because some variations may confer advantages in survival and reproduction. Sorry. We have the mutation, we have the natural selection that Darwin already spoke about. We have the gene flow, which gene flow means randomness. We have the genetic drift means randomness. Gene flow means the migration. We gave the definition of all of these before. Gene flow is the transfer of genetic material a new allele will produce in the population. Species, we spoke about them. We spoke about, okay. I think we spoke about only the micro evolution. We didn't speak about anything else. So the micro evolution, if you want, you can go to all sessions. We spoke about them. Now let's start with the macro evolution. So we have kinds of evolution. We have the microevolution. We have the macroevolution. The microevolution are natural selection, genetic drift, gene flow, mutations. These are microevolution. Macroevolution means large-scale evolutions like speciation, adaptive radiation, divergent, third kind is the divergent evolution. And fourth kind is the convergent evolution. And fifth kind is the co-evolution. Sixth kind is the parallel evolution. And These are the kinds of evolution. We have the microevolution that we already spoke about. We have the macroevolution. We have divergent, convergent, parallel, and coevolution. Let's speak about each one of them before we go to the theories of evolution. So now today we will speak about evolution and in this hour until 5, and we will speak about junctions of the body and about drugs and about smoking. Okay, so evolution, junctions, drugs, smoking. Tomorrow we will speak about naming in chemistry, Le Chatellier's principle in chemistry, and anatomy in biology, and the eyes physiology and anatomy in biology. So tomorrow we have these three, and today we have evolution, junctions, drugs, smoking. Okay. So we said we have these kinds of evolution. Let me repeat some things about the microevolution if you forgot, and continue. So give me just a minute to drink water. I'm coming. Okay, perfect. So, who's with me in the class? Sad, proud of you, Sad. There's no one, only you here. Bravo. So let's speak about. I will finish on evolution. I want everyone to be in the class. No one is entering, so I'll wait until tomorrow. I will finish everything else tomorrow. Today I will just speak about evolution and finish for today. And tomorrow we continue everything else. I don't think they will take more than 3 hours. Tomorrow's class will be after 3 o'clock. Will be like from 5 to 9, something like this. It will be at night. After tomorrow, the first session um, of solving, and then we'll stay solving until the 5th of September. Okay. So, Saad, let's see. So, what are the, we were here in the microevolution. Microevolution is the small-scale evolutionary changes within a species or population over a relatively short period. These changes occur through mechanisms like natural selection, genetic drift, mutation, and gene flow. Examples of microevolution is the development of antibiotic resistance in bacteria or changes in the color of peppered moth during the Industrial Revolution. Okay. Mechanisms, we have the natural selection. Natural selection are certain traits become more common based on environmental pressures. Genetic drift, random changes in allele frequencies, especially in small populations. Gene flow, introduction of new alleles from other populations, and the mutation, introduction of new genetic variations. Macroevolution refers to large-scale evolutionary changes that result in the formation of a new species or groups of species. It encompasses the grand scale of evolutionary changes, including the origin of a new species, mass extinctions, and the evolution of major adaptations. Examples, the evolution of mammals from reptile-like ancestors, the diversification of flower plants like angiosperms. Mechanism, speciation, the process of forming a new species through reproductive isolation. Adaptive radiation, the rapid evolution of diversely adapted species from a common ancestor, often in response to the availability of new ecological niches. 3. Divergent evolution occurs when two or more sharing a common ancestor become more different over time. So two or more species will be sharing a common ancestor. This is called divergent evolution. It's often due to different environmental pressures or geographical isolation. Example of divergent, the evolution of the different species of Darwin's finches on the Galapagos Islands, each adapted to different types of food sources, but all of them come from the same ancestor. Another example of divergent evolution, the differences between the limbs of mammals, such as the wings of bats, the flippers of whales, the arms of humans. So these differences, all of us come from the same thing that we all have arms, we all have hands, but mammals have kind of hands, cats have kind, dogs have kind, rabbits have kind, we have kind. All of us come from having hand, but each one of us has a different one. Mechanisms, geographical isolation, when populations of a species become physically separated, leading to the divergent evolution. And environmental pressures, different environments may favor different traits, leading to divergence. However, the convergent evolution occurs when unrelated species have the similar trait independently. As an example, the wings of bats and birds. Both of them, bats are a species, birds are a species, but both of them have wings. This is called the divergent. Or let's say the body shape of dolphin and shark. Dolphins are mammals, sharks are fish, but each of them from a species, but they have the same trait. The mechanism is the natural selection. Natural selection, similar environmental challenges can lead to the evolution of similar adaptation in unrelated species. Coevolution is the process by which two or more species reciprocally affect each other's evolution. Okay? Example, the evolution of long beaks in hummingbirds and long-tubed flowers, where each has adapted to the other's characteristics. Means when one of the means two species, plants, animals, whatever. When two species try to adapt their shape according to each other's, usually hummingbirds, they have a long beak, so they need a long stigma to start eating on it. So the flower itself made during time, during decades in the evolution process, it made her beak, her stigma to be longer for this hummingbird because the hummingbird have a longer beak. So they try to make a kind of combination for them, both of them, to be able to adapt in the surrounding. Another example of co-evolution, the arms race between predators and the prey. Predators evolve more efficient ways to catch prey, and the prey has more defenses. Let's say the wolf and the dog. Wolves usually have more, they know more how to defend themselves. However, the dogs know more how to catch prey. Mechanisms, we have the mutualistic relationship. Species that depend on each other for survival often evolve traits that benefit each other. So mutualistic relationship means both of them benefit each other. However, the antagonistic relationship, one of them benefit and the other will be dead, like the predator and the prey. This is called the antagonistic relationship. However, the mutualistic, both of them benefit, like the hummingbird and the flower. And the last one is the parallel evolution occurs when two related species evolve similar trait independently, often due to similar environmental pressures, despite being separated by geographical or other barriers. So we have two species have the similar traits because they have the same environment. Examples, the similar evolution of the marsupial mammals in Australia and the placental mammals in other parts of the world. Both of them are evolving similar trait, but each one of them is living in a place. So different. So parallel means different geography but same trait. Okay. Okay. Co-evolution, we have mutualism, means means a good relation between them. We have antagonism, like one of them benefit and the other does not benefit. Okay. So let's say the development of resistance to the same pesticide in different species of insects. That is a parallel evolution. Why parallel? Because all of these species are resisting to pesticide, but this pesticide is this pesticide is thrown on different geographical areas of them. Let's say corn plants in Australia and corn plants in Asia. Each one of them is in a place, but this pesticide was able to adapt to work on both of them, to activate both of them. Mechanism, they have similar selective pressures. Okay, now let's speak about the theories of evolution. Don't worry, I will send on the group what I was speaking now about evolution because I didn't really write so much, and I think you would want to have some notes about them. So theories of evolution.
Ma'am, do we have same geographic location and coevolution? Same diversion?
Yeah, you have same geographical location. Yeah. Is it divergent? Not the divergent, the convergent. The ones who have same geographical location, which live in the same one, and they have different traits from each others. These are called allopatric species. Let me speak about them if you want before we continue. So we have the sympatric species and we have the allopatric species. The sympatric means they are living in the same geographical area. Okay, and they are the same species. However, the allopatric, they are living in different. No, wait. We just make sure of them. Yeah. Okay. So allopatric species is when the when a population is geographically isolated from other populations, okay, of same species. Okay, so like lions that usually live in the forest, they go live in the mountains. This is called allopatric species, means when the same species, they go live in different places. However, the sympatric species occurs without geographical separation. So in this type, we have a new species arise within the same geographic area. So two species living the same geography, but they give a new species. As an example, in East African lakes, fish has diversified into numerous species with different feeding habits and colors despite living in the same lake. So more than a kind and color of the fish are living in the same lake, but they are different. However, the allopatric species, the same species, but living different geographically. So allopatric means different, sympatric means same. But same what? Same species. So here we speak about the species. No, here we're speaking. Yeah, here speak about the species exactly. So because we're saying the species are the same but living in different. So allopatric is same species, different geography. Sympatric we have same geography, different species. In this way. Okay. Now, let's speak of the theories. Where are the theories? Okay. Okay. So, evolution theories. We have Darwinian evolution. We have Lamarckian evolution. We have neo-Darwinism or modern synthesis. And we have punctuated equilibrium. So, what does Darwin say? What does Lamarck say? The main one of Darwin, the main conclusion of this evolutionary theory is the natural selection. Okay, so Darwin, in your mind, put Darwin natural selection. So Darwinian evolution, or let's say the natural selection evolution. Charles Darwin proposed that natural selection is the primary mechanism of evolution. According to Darwin's theory, according to Darwin's theory, individuals with traits better suited to their environment are more likely to survive and reproduce, passing those traits to the next generation. So he says, according to natural selection, the people or the individuals that are more adapting to the environment around them, they have the highest ability or the probability to reproduce and survive. They have so much things that he spoke about. Darwin, I will leave them for the end because Darwin is the most important and Lamarck is the most important. So the Lamarck and Darwin, I will speak about them in general and then I will speak about them in specific. So, Lamarckian evolution, Jean-Baptiste Lamarck proposed that organisms can pass on traits acquired during their lifetime to their offspring. Although this idea has been largely discredited, it was an early attempt to explain evolutionary change. So he just said that they can pass on traits from one organism to the other during their lifetime. It does not start at birth, it starts at lifetime. A person, let's say what's called a giraffe. Yeah, so imagine a giraffe. He said that it might have, when it first was born, it had short neck, and then because she used to eat from the trees, so she had to develop her neck and become long, like tall neck. So he said that he's speaking, although it's super discredited because it's genetically, it must be genetically like this, but he proposed that we might like have, depending on what we lived in our lifetime, we might been like this. We might change our shape this much, our organs this much, just to be able to adapt to the surrounding. The neo-Darwinism, the modern synthesis, is the unifying theory that combines Darwin and the Mendelian genetics together. Okay, to Mendel. It incorporates the role of mutation, genetic drift, and gene flow in addition to the natural selection. This is the modern, which speaks about the microevolution. Punctuated equilibrium proposed by Stephen Gould and Niels Eldredge This theory suggests that species remain relatively stable for long periods and then undergo rapid evolutionary changes during short periods of time, often associated with speciation events. So it says, okay, like long period of their life, they stayed constant, they remain stable, the species. However, on a very small and rapid changes happen, or the punctuation happen in a very short period of time, which might change the whole species and will make a huge event in this species. This is the punctuated equilibrium. Okay, so now let's speak about Darwinian more. Before we end the session. Darwin said, this for memorizing. Individuals, organisms vary in traits they possess. Number 2, some trait differences are heritable. Means evolution, change in allele frequency over time. So some trait differences are heritable. 3, he said, survival and reproductive success are highly variable. He also said individuals with certain heritable traits are more likely to survive or reproduce. He also said natural resources are limited. This is so important to memorize. And he also said the reason of mutation is randomness and not the need to adapt, not the need to adapt. These 6 postulates you should memorize, super important, especially the 5 and the 6. Okay, memorize these postulates. Now I will send you on the group. So like if you want to study. Okay. Okay, guys, I'm done. If you want, you can leave. I'm gonna just send on the group, okay?